I'm a neuropathologist. I work at the Department of Neuropathology of the University of Amsterdam. As a neuropathologist, we have the great opportunity to study really well-characterized clinical material, which is obtained or autopsy or a surgery from patients with pharmacological intractable epilepsy. Studying this material, we can get some information concerning the pathogenesis or the origin of the disease, but also the mechanisms which are responsible for the development of seizure, progression of seizures. And we can use this information to confirm the experimental data and to translate this data in the direction of the patients. So I see neuropathology, which is my field, as a bridge of a link between the basic science and the clinical research. In my group, we are particularly interested in focal lesions, which are indeed target of surgical resection, and particularly the focal malformation of cortical development. These include a large spectrum of disorders and are major cause of epilepsy in the pediatric population of patients, so in children. There are a number of lesions which are summarized here, so the cortical tuber in patients with tuberous sclerosis, of the focal cortical dysplasia, which is common in a pediatric population with epilepsy. And they are different entities, but actually histopathologically they have also some common features. For example, the presence of these large abnormal cells, neurons, but also immature cells. You see here these large eosinophilic cells, which you see in focal cortical dysplasia subtypes and in cortical tubers. And we have also the opportunity with the development of prenatal diagnosis to analyze these lesions, these developmental lesions early during their development, in the early stage of corticogenesis. I show here a tuber which is detected 20 weeks, and then we see 23 weeks the cortical tubers with the developing cortex. So we can follow these lesions from fetal brain to adult brain. And this slide summarizes the key issue concerning malformation of cortical development. So we would like to know how are these different focal malformations formed. So when the malformative process begins during corticogenesis, can we identify the origin of these large cells that I just showed to you, and can we identify common pathogenetic target, which could be or can be target of therapy? And then clearly for us, working on epilepsy, it's important to understand the relationship to epilepsy. So why they are epileptogenic, why they are highly epileptogenic, what is the role of the lesion versus the perilesional brain, and important also here to identify common epileptogenetic pathway. We particularly focus on the mTOR pathway, which is a pathway important during development and relate also to epileptogenesis. And between the epileptogenetic pathway, we provide some evidence concerning the role of the deregulation of GABA receptor transmission, but also the role of inflammation. And for the future, it's really important to understand also the relationship between this malformation, epilepsy, and the cognitive abnormality in the pediatric population of patients. This is a slide which summarizes the idea that we have on the basis of the human study, together with experimental study, that inflammation may play a role in epilepsy, involve different types of cells, glial cells, neuron cells, which through different pathways may contribute to change or to increase in neuronal excitability. And once that you know the pathway, then you can try to target this pathway in experimental model, try to find a better therapy for the patients. So in conclusion, the study of the human tissue with some limitation may help us to get some insight in pathogenesis, epileptogenesis, in pharmacoresistance, why there is this resistance to pharmacological treatment. And we can use this information to better classify this malformation on the basis of specific pathway, to detect this pathway, to modulate this pathway as a new therapeutical strategy, but also to generate a new idea for experimental model that can be used to further analyze the mechanism of epilepsy and the possible therapy.